Morning, everybody. This is another lecture preview. We've had a good snow day. Today is February the 1st. This is a lecture preview coming after cellular organization. We'll have this lecture roughly next week, and then we will wrap it up and then have our first exam. So we'll begin with just some basic terms. Cell division is going to be the process by which cells reproduce themselves. If you guys can remember back to cell theory, the tenets of human life, we talked about how you had an initial cell, and then the cell would divide to produce two cells. Those two cells would then divide to produce four cells. We're going to talk about cell division and how cells can divide, whether they are somatic cells. Somatic cells are going to be anything other than a germ cell. Germ cells are going to be the gametes. This is going to be either your sperm or your egg, so the spermatocyte or the oocyte. And these cells, if they're somatic cells, they'll divide using something called mitosis, which is going to be genetic duplication, and you're going to increase the number of cells. And then meiosis actually cuts down the number of cells in order to produce those gametes, or the egg and the sperm. So again, just definitions: somatic cell, this is any cell of the body other than the germ cell. And I want you guys to know that the term germ cell just means either sperm or egg. So somatic cells undergo something called nuclear division, in which the nucleus divides, and it divides called mitosis. So nuclear division occurs when the cell divides. We call this mitosis. And when the cytoplasm divides, we call this cytokinesis. And those are two important terms, cytokinesis being the division of the cytoplasm, and mitosis being nuclear division. Germ cells undergo two specialized divisions called meiosis. So there's meiosis 1 and meiosis 2, and we'll talk about some important things that occur during meiosis 1 and meiosis 2. Meiosis is going to be, again, responsible for the production of gametes. And it's important to know that whenever you are undergoing meiosis, that there is something called genetic variation, in which the genes are becoming varied, right? They are maybe recombination of different genes in order for us to have something called heterogeneity, or to appear different. So somatic cell division, this is the cell cycle. We've talked about this previously. Again, this is the same concept as if I cut my finger, and how does the cut on my finger heal? Well, it does it through mitosis, through the cell cycle. There's two different phases of the cell cycle. There is interphase. An interphase is going to consist of three different phases. And once we go through interphase, in which the genetic material is duplicating and becoming more and more in order for us to go through mitosis, we will then go through mitosis. And you can remember mitosis through the mnemonic PMAT: prophase, metaphase, anaphase, and telophase. And I'll tell you the important things to know about each of these phases. So again, the cell cycle consists of interphase, and it consists of mitosis. During interphase, we are having an increase in the genetic material, an increase, this is where actually DNA synthesis happens. We're collecting all of the things in order for this DNA synthesis to occur. We're collecting phosphate residues, nitrogenous bases, and sugars in order to produce that DNA base pair. Once we have enough DNA base pairs, we'll then begin to synthesize it. So this is just another picture here showing you interphase. This is divided up into the G1, S, and the G2 phase. So I want you to know that during the G1 phase, this is your initial growth. So we have an increase in growth and normal metabolic roles. Once we go through G1, we'll go through the S phase, and this is where synthesis occurs, DNA synthesis. And go after we have enough, once we begin to replicate and produce the strands, we'll then go on to the G2 phase, which is just another growth phase. So it's important to know the three different phases of interphase, knowing that G1 occurs first, then we go to the S phase, then we go to the G2 phase. And once we have duplicated our genetic material, we can then go through the mitotic phase and prepare for nuclear division. So mitosis, we'll read the text here. Humans are made up of 23 pairs of chromosomes, 46 total. This is called diploid, right? Because somatic cells have two sets of chromosomes, we call them diploid. Two chromosomes that make up each pair are termed homologous chromosomes. They're homologous. That means they're the same pair of chromosomes. During mitosis, after interphase, again, remember interphase is the G1, the S, and the G2 phase. We have a duplication of the chromosomes. They appear, they line up. This occurs during metaphase. Then they separate and finally become the genetic material for the two new cells. So what's important on this slide? Mitosis, knowing that humans are made up of 46 chromosomes. Whenever we have 46 chromosomes, that term is called diploid. We have 23 pair, so 23 times 2 is 46, and those pair of chromosomes are called homologous chromosomes, which means they're the same chromosome. So we could have homologous chromosomes for blonde hair or for hair in general. Here is just a picture of a karyotype. You guys will see here that we have 22 autosomes. This is important. Autosomes are any chromosome that does not deal with gametes. And sex chromosomes are what makes us either male or female. So, for example, this karyotype here has 22 autosomes, and then they have an XY. So, is this a male or a female? That's right, this is a male, absolutely. So these are homologous chromosomes here. As you can see, these two chromosomes are genetically the same. A locus is just where genes are present on a chromosome. Here, this is a locus for the flower colored gene, and it's showing you here the homologous chromosomes. This is the allele for the white flower. This is the allele for the purple flower. And again, these chromosomes, they can either be autosomal dominant, autosomal recessive, or they can be heterozygous. Autosomal dominant, that means if one chromosome here is dominant, that means that will be expressed. If they're both autosomal recessive, then they will express the recessive phenotype. But again, if you have a dominant gene, that means the dominant phenotype will be expressed. And again, heterozygous, the dominant gene will be expressed. The dominant gene always is the expressed gene, and the recessive is only expressed when there's the presence of two recessive alleles. So going back, just to kind of bring everything that we've discussed to a summary, we've talked about how mitosis is termed nuclear division, right? Meiosis is going to be the production of gametes. Mitosis, or the cell cycle, is consistent of two different phases. We have interphase, which is going to be the increase in genetic material. This occurs through three different phases. G1, which is the initial growth phase. S phase, which is where DNA synthesis occurs. And then G2, which is where the second portion of the growth phase occurs. And again, all of this cell cycle occurs within what organelle? The nucleus, absolutely. DNA synthesis occurs within the nucleus. That's important to know. And once we have completed interphase, we then go on to what is termed mitosis. And now we're going to get into the mitotic steps. You can remember the steps of mitosis through the mnemonic PMAT, where you have prophase, metaphase, anaphase, and then telophase. So I want you to think of prophase as being the chromosome or chromatin condensation. So chromatin, if you guys remember, chromatin is just going to be where you have the DNA strand associated with a protein. So this chromatin is then going to condense, meaning get smaller. And chromosomes are going to be held together by a centromere. I'll show you that on this next portion. A centromere is just going to be a portion in the middle of the chromatin that is going to essentially hold the chromosome together. So you have a kinetochore and a mitotic spindle that appears as well. We'll show you that. This is the chromatid right here. whole thing is called a chromatin. This is going to be the centromere. The centromere is just the portion in here that holds the chromatid together. And again, does anybody remember what the centromere is made of? It's on this slide, microtubules. And why do you think that's important that it's made of microtubules? Because microtubules are responsible for movement. So as you can see, as these chromatids begin to move, you will see that the microtubules are going to be very important for that. So again, just showing you here, prophase, this is where chromatin fibers condense and become visible. So they condense and they actually become visible in this stage. We say that here that chromosomes are held together by a centromere. During metaphase, I want you to think metaphase as middle. So the microtubules of the mitotic spindle, remember at the centromere, this is going to cause them to line up at the metaphase plate in the middle of the cell. Anaphase, A means apart. This is where the centromeres split, pulling the two members of each chromatid pair apart. So the actual centromere, this will cause the chromatids to separate to opposite sides. So as you can actually see this in underneath the microscope, you can see the chromatids appearing at different poles. During telophase, this is where the separated chromosomes are now at the opposite ends. This is where you will have the chromosomes appearing at opposite ends of the cell, and they unwind this and they begin to revert back to the chromatin fibers. I think it's really important to know that chromatin condensation occurs during prophase. The chromosomes line up in the middle of the cell during metaphase, and anaphase is when the centromeres split and they're at opposite poles. Cytokinesis is going to be the formation of a cleavage furrow. This is whenever you actually have division of the cytoplasm itself, and it splits it into two new daughter cells. Here's a picture that might kind of bring this into, might kind of help show you what we're talking about. This is chromosomes consisting of two sister chromatids right here. Condensation is going to occur, and you're actually going to be able to see the chromatids themselves. This is formation of the early mitotic spindle, which is going to be responsible for movement. We said that has occurred during prophase. During a metaphase, you're going to see the lining up of the chromatids on the metaphyseal plate. Remember, metaphase means middle. Anaphase, again, this is the mitotic spindle is going to begin pulling these chromatids apart. They're going to separate to opposite poles right here. Telophase and cytokinesis are going to occur very closely together. Formation of the nuclear envelope, this is very important part during telophase because this nuclear envelope, again, this is going to be responsible later on. We know for protecting the nucleus. We also know it's going to be responsible for protein transport. It's going to allow protein transport out of the nucleus into the rough ER. From the rough ER to then move on to the Golgi. You guys can remember that from the cellular organization lecture. So cytokinesis is this actual division of the cytoplasm where the cleavage furrow is forming, the cytoplasm becomes split, and you have two new daughter cells. Here's just another picture showing you this under an electron microscopy. So meiosis. Meiosis is also referred as reproductive cell division. Again, reproductive, so we're dealing with sperm and eggs, right, or gametes. Follows the same steps as mitosis, but undergoes them twice to reduce the chromosomes number to 23. So you actually have two phases during meiosis. You have meiosis 1 and meiosis 2. So instead of forming a diploid state, as we are in mitosis, we're forming a haploid, or half the number of chromosomes, 23, in meiosis. We say that germ cells here are haploid in number, so that means they're just N or 1N because they only contain half the amount of genetic material of that of mitosis. When two germ cells fuse together during sex, the chromosomes number then becomes 46, bringing rise to a new organism. There's a term called crossing over, and this is a very important part to know. This is what gives us what we call genetic variation, or in other words, to describe it, genetic diversity. This is when when parts of two homologous chromosomes interchange. So again, you've got these homologous chromosomes, which code for a certain type of phenotype. This is whenever they exchange genetic material, and this is what gives us our genetic diversity. Meiosis is, again, this is meiotic division 1 and meiotic division 2. This is, again, just showing you this initial DNA replication and genetic recombination. Here you're going to have crossing over of genes, which they're going to recombine in order to, again, give us this genetic diversity. Again, during the end of meiosis 1, you're having this telophase portion in which you're having the cleavage formation of the cleavage furrow and formation of two daughter cells. During meiosis or meiotic division 2, where again we're beginning to reduce the number of chromosomes that we have. So 2 is coming down here, and this cell right here is beginning to produce 2, and then we're going to wind up with 4 daughter cells. This here is going to show you what crossing over kind of entails. The homologous pairs moves close together. The chromatids may exchange genes. So it's really this exchanging of genes in which we are calling it crossing over. As you can see here, the red and the blue, you've got a crossing over from a red with a blue and a red with a blue. This is what's going to give us our genetic diversity. Again, here are some videos that will kind of help you understand mitosis and meiosis. I think these will be very good videos for you to watch and would kind of help solidify this material. So again, knowing that interphase is going to consist of G1, S, and G2, knowing those steps in order, knowing that this is where the initial genetic duplication occurs. Knowing from there, we go into mitosis. And again, mitosis is for germ cells. Germ cells are any cells that are not, mitosis is for somatic cells, which are any cells that are not germ cells. Meiosis is for germ cells. Meiosis is going to actually reduce the number of genetic material and produce a haploid state. Meiosis has the same number or the same steps of mitosis. Mitosis has prophase, metaphase, anaphase, and telophase. Prophase is where the chromatids begin to condense. They first become visible here. Metaphase is where they line up on the metaphyseal plate. Anaphase is when the mitotic spindle is beginning to pull these chromatids apart and they are on separate poles. Telophase is when they were completely at separate poles. This is where the nuclear envelope is first forming. The nucleolus appears here as well. Once we have formation of the cleavage furrow, we then undergo cytokinesis, which is just cytoplasmic division. Remember that meiosis is for germ cells. We're producing a haploid state, like I previously just said. A haploid state is going to be where there is only 23 chromosomes. And again, meiosis is going to help us produce genetic diversity through crossing over. These are just some important points. If you have any questions, we'll cover this in class. But again, this is just a lecture preview. Thank you guys for tuning in today.